before when I wanted to see it.
Okay, we're recording now.
Oh, okay.
Awesome.
Alright, so this is the Chapter 11 review with Emily Siniscalco, Madeline Arcurio, and Stephanie Coppola. We're going to be doing the test yourself questions at the back of the chapter because we didn't want to brave learning catalytics.
I don't think we could have if we wanted to.
Yeah, I don't think we have a slot.
Well, that and it just wasn't working at all. No one could get on, I think.
Yeah. Well, they were trying to fix the problem on Twitter, but I don't know if anything came of it.
Question numero uno. The function of the extracellular matrix, ECM, in animals is A, to provide strength. Am I in the right chapter?
Wait.
What question are you reading?
What question are you reading? That's not the question I have.
I'm in the wrong chapter. Sorry.
No, you're good.
Question numero uno for real.
Read it louder.
I was on chapter 10. An agent that allows a cell to respond to changes in its environment is termed A, a cell surface receptor, B, an intracellular receptor.
Still the wrong chapter, right, Emily?
11.
Wait, can I just read the question?
Wait, let me find it first.
It's on page 234, I think.
Okay, it starts with why did researchers, right?
Yeah.
Can we start over?
Oh,
no.
I don't know. I've never used this before.
Sorry.
No, it's fine.
Sorry.
Oh. Why did researchers initially believe that genetic material was protein? A. Proteins are more biochemically complex than DNA. B. Proteins are found only in the nucleus, but DNA is found in many areas of the cell. C. Proteins are much larger molecules and can store more information. D. All of the above. E. Both A and C.
Okay, so who wants to take this one?
I think we should start by discussing it.
Well, yeah.
So A. Proteins are more biochemically complex. I think that one is true.
You're really quiet.
I'm really quiet?
Yes.
Yeah, you're a little bit quiet. I turned my volume all the way up and you're still a little quiet.
Wait here, let me see if this is. How about now?
No, that was just clicking.
Okay, I'll just talk louder. So proteins are more biochemically complex than DNA. I think that one's correct because they originally said that proteins have 20 amino acids, whereas DNA only has 4 nucleotides.
I would have to agree with that. Okay. And then by looking at letter B, you can tell that it's not true.
Yeah. DNA is only found in the nucleus, and proteins are found in many areas of the cell. So.
And C is not true either. DNA, you have a lot of DNA. Something like you would have, if you unwound it all, you would have 1 meter of DNA, and it has to fit in a cell that's 10 to 100 micrometers, which is why it's so compacted with the various levels of...
Yeah, so proteins definitely aren't larger, so I think it's just going to be A.
I think it's A, too. Like,
I would have their answer sheet out.
What?
Does anybody have the answers out so we can check? Oh, let's just check them all at the end.
No, I don't have the answers out. I'm writing them down, though.
Here, we can just check them all at the end.
Yeah, okay.
So 1A. Who wants to read 2?
Considering the components of a nucleotide, what compound is always different when comparing nucleotides, or what component is always different when comparing nucleotides in a DNA strand or an RNA strand? For this one, I said B, the pentose sugar, because it says which component is always different.
Always different.
Yeah.
I agree. That's what I was
pentose sugar in RNA is always ribose, which has one more hydroxyl group than deoxyribose.
Oxygen group. And the nitrogenous base is different sometimes because of uracil, but it's not always different.
Wait, but guys, the question, it says when comparing nucleotides in a DNA strand or an RNA strand, it's not saying and them. So you're not comparing RNA and DNA. You're comparing specific nucleotides within a strand of DNA or within a strand of RNA. So it wouldn't be the pentose sugar. It would have to be the nitrogenous base. Maybe.
No, I think we read it right.
either.
What?
I think it is what we were thinking at first, because the nitrogenous base isn't always different, because you can have two adenine nucleotides. I think it's just weird phrasing.
Yeah, if you compare two adenine nucleotide DNAs, then all components are the same. There's not a component that's different. I think we're reading it right. It's just
I think it's an awkwardly worded question.
Mm-hmm.
Yeah.
I don't know.
But I think it is the question that we first read.
Either way is definitely what it's saying, but I don't think any of the answers could fit that. So I think it's the pentose sugar, and then it's DNA versus RNA.
Steph, your turn.
Okay. 3. Which of the following equations is appropriate when considering DNA-based composition? Percent A plus percent T equals percent G plus percent C. Oh, wait, do I have to read all the options?
No, just pick the one you
Okay,
chose.
let me just look at them. Wait, sorry. I think it's A plus T. Wait, what?
I was just explaining. Go.
Oh. Sorry, I know how to answer this question. I'm just drawing a blank. I think that it is A plus G equals T plus C, so D.
That's what I said.
I think that one's correct as well, because your amounts of A and T are always going to be the same, because they are paired together. And then your amounts of G and C are always going to be the same, because those are always paired together. So,
yeah.
Of course, A, which is equivalent to T, and G, which is equivalent to C. A plus G equals T plus C.
Yeah.
All right. So, number 4.
Numero cuatro. If the sequence of a segment of DNA is 5' CGCAACTAC 3', what is the appropriate sequence for the opposite strand? So I think this one we're going to want to write out on our paper.
Yeah, probably. I keep going off the screen here.
So there are two components that we have to keep in mind when we're doing this problem. We have the 5' to 3' component, and we have the actual base pairing rules. So we know in base pairing rules, C pairs with G through 3 hydrogen bonds, and T pairs with A through 2 hydrogen bonds. And each one has a pair composed of a purine and a pyrimidine. And then the strands run anti-parallel, which means that a 5' to 3' strand would be on top if this is the 5' end and this is the 3' end. And then the complementary strand, this would be the 3' end and this would be the 5' end on the DNA strands that are my arms.
Yes.
So if this strand is 5' to 3', then we know that the complementary strand must be 3' to 5'. So that way we can eliminate A and C right away, and then we only have B and D left. And then we just follow the base pairing rules to see which one is the actual match for the base pairs. And you can do that by looking at the very first nucleotide there.
Well, yeah, and then even like that, you could also eliminate B and C immediately, because if you look at the original strand, both ends end with C, and C does not pair with A.
It pairs with
G. So you know that it's not B or C.
Mm-hmm.
So
the answer is D.
D. We all agree on D?
Mm-hmm.
Awesome.
All right, so number 5. Of the following statements, which is correct when considering the process of DNA replication? So our choices include new DNA molecules are composed of 2 completely new strands. New DNA molecules.
What?
Do we want to go one by one?
Speak up.
Do we want to go through them one by one?
Okay.
We can. Okay. So
new DNA molecules are composed of 2 completely new strands, and that would be the. Ainsley took my pen.
This is not your pen.
No, this is my pen.
What?
Wait, this isn't my pen. She gave me a different one. Okay. Sorry. So, new DNA molecules are composed of 2 completely new strands. That's not true. Because that would be of the conservative... That would be of the... Sorry. That doesn't precisely fit of any of the models. No.
Wait, wouldn't that be the dispersive? I don't know.
I don't think that would be...
No, that would be just
That would be
wrong.
Like,
That would just be wrong.
Yeah, that's not a particular model, really.
You just can't have it completely.
But
the dispersive is they are composed of like fragments of each one, like all mixed together.
But they technically called them new strands. So I think that's what they meant by new. I don't know.
Yeah, they technically be called new strands, but it wouldn't be entirely new DNA.
Yeah. No, I get that part.
A little bit of old, and then
one is just wrong because you have even with the conservative, when you have this one that's made of 2 new strands, you have another one. Well, the conservative model is wrong. So let's move on to B.
Okay, so letter B is the one that I put as correct because it says new DNA molecules are composed of 1 strand from the old molecule and 1 new strand, which we know to be correct when we look at DNA replication because the 2 strands of DNA are unzipped and then each one has a new strand built off of it.
Yeah, the semi-conservative model.
I agree with that because of Melselin and Stahl's experiment. Sorry if I pronounced that first name miserably wrong, but that
I can't even hear you, so
yeah.
I agree with that because of Melselin and Stahl's experiment where they determined that it was semi-conservative through the use of heavy and light nitrogen isotopes and centrifuge.
I think what this question is asking specifically is when you think about how DNA is replicated, how does that fit with like the semi-conservative model?
Yeah, I know. I'm just saying that the semi-conservative model was proved by their experiment.
Through that
so.
Yeah.
I guess
which moves us to number 6.
Yeah, I was just looking down about to say that. And I guess if we decided that B is the answer, we don't have to go over C and D. So.
Hey, look at that.
You're on top of the game, Maddy.
Selson. I think it's Stephanie's turn.
Okay. Great, now I get to pronounce his name. Okay.
I think it's Messelson.
Messelson, I think so. Messelson and Stahl were able to demonstrate semi-conservative replication in E. coli by A. Using radioactive isotopes of phosphorus to label the old strands and visually determining the relationship of old and new DNA strands. B. Using different enzymes to eliminate old strands from DNA. C. Using isotopes of nitrogen to label the DNA and determining the relationship of old and new DNA strands by density differences of the new molecules. And D. Labeling viral DNA before it was incorporated into a bacterial cell and visually determining the location of the DNA after centrifugation. Centrifugation? That's like a weird word to pronounce.
That was not correct anyway.
What? Well, it's not true. It's not correct anyway, no. C. Using isotopes of nitrogen to label DNA and determining the relationship of old and new DNA strands by density differences of the new molecules.
Yep.
I agree with that. And it was because I feel like we should explain them.
Yeah, we have to.
It was because they used the nitrogen 14, which was the light nitrogen and the more common
And the nitrogen, 15. Yeah, louder.
Oh, louder. Okay.
Yeah.
It was because they used
correct.
the
You need to speak up, okay?
So they put an E. coli bacteria. I feel like I'm screaming. They put an E. coli bacteria into a solution containing nitrogen 15, and they let them divide and grow in that solution, so that all of their original DNA, or the DNA that was present at the beginning of the second stage of the experiment, contained the nitrogen 15 or the heavy nitrogen. And then they moved these bacteria into another solution that contained nitrogen 14, and once DNA replication occurred within this new solution, the new strands would definitely all contain the nitrogen 14 instead of the nitrogen 15, and then after the first round of DNA replication, they took the genetic material from the bacteria and they put it in a centrifuge. So the light nitrogen should be on the top and the heavy nitrogen should be on the bottom, and you can distinguish between new and old strands. And that was how they determined the semi-conservative model was correct. It's very hard to talk with my hands when I have to keep the microphone right in front of my face.
Was the heavy DNA on the top or the bottom? I thought he said that the heavy DNA was on the top.
Well, it's a centrifuge, so it would be on the bottom because it's
because of gravity.
Yeah, I guess.
I think it's confusing with the picture because as the generations go on, the top bar gets darker, but that doesn't mean heavier. It just means that there's a bigger population of that DNA.
Yeah. I think I just copied it into my notes wrong.
Yeah, so like if you have liquidy stuff and then you have rocks, if you put them in the rocks would end up on the bottom. That's my thing.
We can't see your notes.
We see a rock. It's okay. We get the concept.
Yes. We got it, Maddie.
Maddie.
I think it's your turn to read the question, Maddie.
Oh. During replication of a DNA molecule, the daughter strands are not produced in exactly the same manner. One strand, the leading strand, is made towards the replication fork, while the lagging strand is made in fragments in the opposite direction. The difference in the synthesis of the two strands is the result of which of the following? A. DNA polymerase is not efficient enough to make two good strands of DNA.
That's awfully subjective.
I feel like it's more of an opinion.
No, it's not.
What?
The answer.
What? Wait, Emily, what'd you say?
I said, no, it's not. A good strand of DNA is one that doesn't give you a terminal disease.
Wait,
and also
wait. I said that's subjective. I feel like that's a subjective opinion.
Of what a good strand of DNA is? Or that it's not sufficient enough? Or efficient enough?
Okay, we've all established that A isn't the answer.
Yeah, but I just said that choice was very subjective and not true.
I agree with Stephanie.
Thank you.
The two template strands are anti-parallel, and DNA polymerase makes DNA only in the 5' to 3' direction.
I vote for that one.
I vote for that one as well.
Because it's not caused a breakage of the DNA strand by UV light.
And I think the cell has enough nucleotides to go around.
Nucleotides. We got enough nucleotides.
Yeah, we have enough nucleotides. So, yeah, the two template strands are anti-parallel, and DNA polymerase makes DNA only in the 5 to 3 direction.
Because it reads it 3 to 5 because it needs a hydroxyl to build off of, which we know.
Yep. And then also just if you have two replication forks, the directions are opposite, so one has to like open the DNA so that it can attach the primer, and then it has to do it in small fragments rather than one continuous leading strand.
3' to 5', so our happy little
Speak up, Maddie.
And it goes from 3' to 5', so the happy little nucleotides can their hydrogen bonds match up with where the partial charges are on opposite strands.
Yeah.
So I guess
you're so quiet.
That's just explaining why they're anti-parallel. I'm actually screaming and Ainsley is laughing at me.
We're laughing at you too.
She just yelled back at me that she's not laughing.
Tell Ainsley we said hi.
Oh my gosh, you're so quiet. Okay, number 8. In eukaryotic cells, chromosomes consist of DNA and RNA, DNA only, RNA and proteins, DNA and proteins, or RNA only.
Yeah, I vote D.
I vote for P too, Maddie. See, DNA and proteins. As we know, chromosomes consist of DNA and histone proteins. They're wrapped around 8 histone proteins in order to make it compact and in order to fit this 1 meter long strand of DNA into a 10 to 100 micrometer cell and an even smaller nucleus.
Yep. Okay, number
And
9.
Further into a 2 to 4 micrometer nucleus. Did you already say that?
I said even smaller, but 2 to 4 works. Now don't go too much deeper because number 9 does that for us.
And can I explain the next one?
Yes, you may.
Yay. Okay, so number 9. A nucleosome is a dark staining body composed of RNA and proteins found in the nucleus, a protein that helps organize the structure of chromosomes. Another word for a chromosome, a structure composed of DNA wrapped around 8 histones and the short arm of a chromosome. So I say D again, a structure composed of DNA wrapped around 8 histones. And it's the first level of DNA folding so that it can tightly wind itself into chromosomes. So,
Yep.
It does have a function. Otherwise, it would be a meter long of DNA in a small cell that's only 10 to 100 micrometers. And that just wouldn't work.
Yeah, so you have all these really, really positive histones, positively charged.
Wait, speak up.
You have all these really positive histones, positively charged, and then the negatively charged DNA is okay, here we go, let's wrap around this. And then it gets really compact and decreases in size 7-fold, so you can start wow, that was starts. You can start to get it to the size that it needs to be to fit inside the nucleus.
Yeah.
Yep.
Awesome spaghetti.
Maddy, your turn to read.
My turn. The conversion of euchromatin into heterochromatin involves A, the formation of more nucleosomes, B, the formation of less nucleosomes, C, a greater compaction of loop domains, D, a lesser compaction of loop domains, and E, both A and C.
I vote A and C. So option E. I don't know if you guys, what do you think? But I'm not necessarily sure if you can have more nucleosomes, but if you could, then I vote that as an answer.
I think it's just C.
For some reason, I feel this might sound ridiculous, but I feel like I remember when we were reading it, it said that the loop domains get, that in heterochromatin, the loop domains are more compact.
Yeah. Mm-hmm. Yeah, we were both saying that.
Yeah.
Oh, yeah, that is what we were both saying. Sorry, for some reason I read the question as backwards, and I was like, I don't think so.
Our only question, I think our question is whether you can have more nucleosomes or not.
I think I'm pretty sure it's just the loop domain.
Maddy, what'd you say?
I just said I agree. I think it's the C.
Just C?
Yeah. Because I don't know if the cell would be able to make more nucleosomes, but it specifically says that it can make more loop domains, and well, not more loop domains.
Yeah.
It can compact these loop domains even further, so that when they ravel all around, you get a smaller chromosome, and then mitosis.
Okay, so C.
Yep.
Yeah, I think so.
Okay.
On to the conceptual questions.
Oh wow! Look at that.
So what are the 4 key characteristics of genetic material? What was Frederick Griffith's contribution to the study of DNA, and why was it so important?
So.
Okay. Well, the first. Oh, what?
Oh, go ahead.
Oh, okay. Well, the first key characteristic of genetic material is information. It has to be able to carry the genetic information that codes for all cell replication and all the functions of the cell. So it has to be able to store that.
And how does DNA do that?
In the nucleotide sequences. I don't know.
Yeah, nucleotide sequences, which are read by RNA polymerase to make
mRNA.
RNA. Just cracking.
What's that clicking?
I don't know.
Maddy.
Maddy?
I was turning on the headphones.
And then that's converted to protein, which is how it's expressed in the cell.
And then
the next key feature is replication. Are you talking? No.
Oh, I was just going to say it, but you can go next. I'll take the next one.
Sorry, you're so quiet. Is replication. And this is important because we know that our cells have to divide in order for us to have living cells, because cells have a finite lifespan and it's often much shorter than our lifespan. So they have to keep dividing in order for us to have healthy tissues. In order for that to happen, the DNA of the cell that's dividing needs to be replicated so that each new cell that's formed can have a complete genome.
And then And then the third characteristic of genetic material is that it has to be transmission. You have to be able to get it from one organism to that organism's offspring.
Yeah. Because otherwise you won't have offspring.
Yeah, or else you won't have more organisms, and that would be an awful species.
Yeah. And then the last characteristic of genetic material is variation. The genetic material needs to be able to change so that organisms can respond to their, can evolve in their, environment. So you have, evolution, and that they can survive and have adaptations that allow them to better live. So. And also,
if you can do the and also
the same, then one event could wipe out an entire population. Yeah. Sometimes that's seen in organisms who have asexual reproduction, where they just
reproduction.
Oh. Yeah, Maddy, what'd you say?
Oh, I just said budding, and then I said asexual reproduction. And
we're remembering the same lab discussion.
Yes. Okie dokie, so what was Frederick Griffith's contribution to the study of DNA, and why was it so important? So, Frederick Griffith determined what was... sorry, I cannot talk today. So, Frederick Griffith, basically, he determined what the genetic... Okay.
I think he determined that there was genetic material.
Mm-hmm. Determined that
they didn't really know that it was DNA yet, but that there was this material in this factor in in bacteria, in cells, that coded for its functions and that could be passed to another organism.
I think they already knew that genetic material existed. What he did was discover that transformation can occur.
Yeah.
Which is when genetic material from one cell goes to another cell.
Yeah, that can be transferred.
And he did that with his experiment with rats, where he was studying the Streptococcus pneumoniae. Wow, that was awful pronunciation, but it might have been right.
Sorry, we can focus
pneumonia.
Wait, you can't hear me at all?
No, we can. It's just really quiet.
It wasn't happening. Okay. I think I was talking over you.
So he had these mice, and he had these. There's two different strains of the Streptococcus pneumoniae bacteria, and it one of which has a smooth capsule, and it's called the S strain because it's smooth, and it has this capsule, and that protects it from the host defenses, so it can infect the organism with pneumonia or meningitis when it gets into that organism's body.
And it will be.
Then the other type is the rough strain. And sorry, I'm screaming. I can't hear anything you say. You're sorry. So, and then there was this other strain called the R strain, which had no capsule, so it looked rough, and that is why it was called R, because it looked rough. And so what he did is he took a S strain and he puts it into a rat, and the rat dies. And then he puts an R strain and he puts it into the rat, and the rat doesn't die. And then he kills an S strain with heat. So that S strain is dead. not functional mouse lives. But then he takes an S strain that's heat killed, and he puts it in with an R strain. And when that happens, the mouse dies as well. And when they take out its blood, they find that it has living S strain bacteria. And like the S strain bacteria didn't come back to life. What happened was that the genetic material from the R strain was somehow transferred. I mean, the genetic material from the destroyed S strain was somehow transferred to this R strain bacteria, and that made it so it could reproduce these proteins, and in turn, the proteome found within these S strain bacteria, and make it so the R strain has a capsule and isn't susceptible to host defenses, and mouse equals dead.
Yeah, I think
that was my very exuberant explanation.
Yeah.
Okay.
Anything to add? Or
that process, just to clarify, the process of taking up that DNA into another cell and causing the change in the proteome is called transformation in bacteria.
Yeah. And then just why was it so important is the last little bit there. And a
genetic material existed.
Yeah.
And this is the first step into like discovering what it is and how it works, is to prove that it existed and then use this knowledge to build on. Which occurs in chapter. Question 2. The Hershey and Chase experiment used radioactive isotopes to track the DNA and protein of phages as they infected bacterial cells. Explain how this procedure allowed them to determine that DNA is the genetic material of this particular virus. Can I do this one?
Yeah, okay.
All right, hold on, we lost a Maddy. Yay. Okay, so in the Hershey and Chase experiment, what they did was they had a virus, a phage, which was going to infect a bacteria. And this particular type of virus is basically only made out of DNA and proteins. It has a protein coat and like legs and stem-like structure. And then it houses the DNA inside, and it will attach itself to a bacteria and insert the DNA into the bacteria, where the protein coat will stay on the outside. And then the bacterium is then transformed into basically a host for making more of these viruses. What Hershey and Chase did with this was they labeled both the protein and the DNA. They labeled the protein with radioactive sulfur because sulfur is only found in proteins and biological molecules, as we saw in the sulfide groups on cysteine. And then they labeled the DNA with phosphorus, and because there's only phosphorus. Phosphorus is only in DNA in biological molecules. It's also in ANP and other nucleotides, but in this case, we're only.
It's not in proteins for our purposes.
Mm-hmm. So when they observed this, they observed that inside of the cell, you could see a lot of radioactive phosphorus. That's where all the radioactive phosphorus went. And then outside of the cell, there was all the radioactive sulfur. And I believe there was 80% of the radioactive sulfur was still outside of the cell. And then of the bacterium.
Was it 85 or was it 65? Was it 85 or 65?
80 of the sulfur was
outside,
for the phosphorus. 65 of the phosphorus was inside, I believe.
Okay.
The important thing is that the phosphorus went into the cell, and that indicates that whatever contained the phosphorus is what's causing the transformation to happen, to transform this bacteria into this just virus factory. So that would be the genetic material, the hypothesized genetic material. And because we saw that this was labeled with phosphorus, it means that genetic material is DNA.
Yeah.
Anything to add?
Nope, that's good.
I think you covered it all. Okay. 3. A principle of biology is that structure determines function. Discuss how the structure of DNA underlies different aspects of its function.
Oh, well, also sorry for dropping out of the shot so often. The cat is on the table drinking things it's not supposed to be drinking. So I have to move her off the table. Izzy.
Izzy.
Sorry, continue.
Okay. Principle of biology, structure determines function. Discuss how the structure of DNA underlies different aspects of its function. Well, wait, I don't know. Okay. Well, one is that when DNA expands, it's a linear structure and it can be unzipped so that it can bind with all of its nucleotides to replicate.
Yeah, I think the one of the big things is that the nucleotides all have these
they all have specific base pairs.
Well, no, with chapter 2. They can all bind with a specific pair because they have partially charged reasons, and that allows them to. Ooh, someone joined.
Yeah, I noticed that too. I don't know who that is though.
And that allows the nucleotides to
I don't know who they are.
Sorry, that allows the nucleotides, since they have these oppositely charged oppositely partially charged molecules at these different points on their structure, they can interact with only one specific.
They left, I think.
Can join with only this one specific. Sorry, the joining and unjoining of that person very much confuzzled me in terms of.
Yeah, me too. It threw me off.
I'm gonna start over. So, you have these nucleotides, and on the different nucleotides, there's different regions where you have these partial charges. And when you have a partial charge, what happens is it can form a hydrogen bond with an opposite partial charge. And these opposite partial charges are found on various regions of the different nucleotides, and they vary. And this makes it so it can very specifically bind to a part of another nucleotide, making these specific base pairs so you can easily replicate DNA and then be able to use this future stuff with mRNA. Well, future chapters with mRNA being transcribed and then translated, and various helpful things that are caused by the opposite partial charges of the nucleotides.
Mhm. Yeah, and I'm not sure if you covered this. I'm sure that you did, but just the specificity of the bonding between the different pairs of nucleotides allows it to replicate and allows it to identically, so that you can have the same DNA in all of your cells, which is important because this way you can code for all of the cells of a specific tissue can code for the same proteins, and they can have a similar function and work together as a unit. And then what?
I was just going to say the specificity, it also helps with coding for proteins. It allows it to express the same gene in every cell to express the same protein. Not really getting into the splicing and how it can be alternatively spliced in different cells, because that is also a specific process. But it's not. It's
what?
My foot cramped because I do nothing but sit. But
Yeah, that answer was much better than the just joined one I gave. I don't know why another person.
Sorry, you're dysfunctional.
I don't know why another person confused me so much, but.
Do we want to talk about how it can fold up and tightly bind so that it can fit into the cell and so that it can separate and divide easily for some replication?
Yeah, I think the only thing that's specifically with the structure of DNA itself would be that the double helical shape makes it slightly more compact than if it were just two linear
lines. Mm-hmm. What is happening on my screen?
We're supporting each other.
I was about to say, wait, you're not supporting me?
Okay, so now that we've... Oh, nope, there are more questions. Never mind.
Yeah.
Collaborative questions.
Oh, by the
way,
I got out my answers, and all of the answers that we put for 1 through 10 were correct.
So Wait, what did you say?
I got out the end of my textbook, and all the answers we put for 1 through 10 were correct.
Yay!
Okay. And I'm guessing there are questions
lighting for my
1 through 3, and then 1 and 2. We lost an Emily.
Bye, Emily.
The light was blinding me. I couldn't see.
Nice.
I either moved it or I needed to get sunglasses. It was.
I think you should have gone with the sunglasses, but not the ones with the sticker. I knew she was gonna.
Come back. Okay. A trait that some bacterial strains exhibit is resistance to killing by antibiotics. For example, certain strains of bacteria are resistant to the drug tetracycline, whereas other strains are sensitive to this antibiotic. Describe an experiment you would carry out to demonstrate that tetracycline resistance is an inherited trait carried by DNA of the resistant strain. Well, I think we could do something almost like Frederick Griffith's experiment, where we see if the transformation would occur if we made.
Yeah, I feel like that would definitely be a helpful experiment.
Because inherited means it's in the DNA.
Yeah.
Yep.
So the experiment would have something to do with killing the resistant ones and in putting them into a system with the non-resistant ones that are alive and seeing if they will be taken up, and then the non-resistant ones will become resistant.
This is more about the properties of DNA rather than its structure.
Mm-hmm.
And since we know that transformation is one of the things that occurs to DNA with DNA because of Frederick Griffith, we could use this same experiment to.
Or at least a very similar one. Yeah.
How might you provide evidence that DNA is the genetic material in mice?
Avery McLeone and
McLeod.
There's one more.
Oh yeah.
Hold up.
I have everything written down. It's just a question of where.
Avery McLeod and McCarty.
Yes.
Yeah. So you could replicate their experiment to prove that it's the genetic material with the test tube. I guess in mice specifically, though. So.
Because they used the bacteria, they purified the DNA in the bacteria.
Yeah. I guess what you'd have to do is you probably have to take mice cells and then use DNA, RNA, and DNase, RNase, and protease.
Well, I guess, yeah, you'd have to.
So mice cells aren't going to transform like a bacterial cell would have. I don't know if the experiment is really applicable directly in this situation.
I don't know. I mean, I guess if you could isolate a factor of mice's like DNA that was transformable, I guess maybe if you use different tissue cells. I don't know.
Oh,
but that's more of a proteome thing. But like if there was something.
Do you remember when we were learning freshman year about gene expression, and we learned about how you can put some of a genome into a bacterial plasmid?
Oh, yeah.
Produce that. So if you isolate a gene of DNA from a mouse cell, and then you try and put part of its genome into a bacterial cell and see if then the protein it codes for was produced, then you could prove that it's genetic material that you isolated and inserted into the bacteria.
Well, yeah, I think the first step would be isolating a part of the DNA and then tagging it radioisotopes of and then also putting the different enzymes that degrade the different molecules DNA, RNAs, and protease.
Tag it because it follows the same principle as the Griffiths experiment.
I do think you need to use the DNase, RNase, and protease because that was originally to make sure that there's not a residual protein or RNA within this genetic material that you're taking out, because you can't go in and be like, this is solely DNA, which is why. So I think what Emily is saying is correct, but I think you also need the DNase.
Yeah, it has to be like a 2-part experiment. Oh, my cat came to join me. Hi.
I'm the only catless one today. It's funny. I have more cats than all of you.
So many cats. You do have more cats than all of us. Well, actually, no. Maddie has 2 cats, right?
Mhm.
Emily has 5. Emily has 4.
Yeah, I have 2 cats, and then Maddie has 2 cats. So you have the same amount as us combined.
I didn't say combined. I just have more cats than you and more cats than her. But that is not the topic at hand.
I know, it's not the topic at hand. Well, that was the last question.
Yeah, but we weren't done with it.
Okay. But anyway, I think it's like a 2-part thing. Like, I think we first have to determine that there is genetic material, and then, and what it is, the molecule, and then we have to determine whether it's like DNA, RNA, or protein, or a protein.
Because you can't simply isolate DNA. There's always going to be residual, which is what the concept behind the Avery experiment was.
Yeah.
I think you might be misunderstanding why they used the enzymes. Because what they did is they had all the 5 test tubes, and they put the R cells and then the DNA from the S cells in there. And then they put the 3 enzymes in there, and then the only one that transformation didn't happen in was the DNase. So
Yes.
that was just to show that the DNase is the one that's actually causing the change.
Well, I
The
think
DNA is the one that's actually causing the change.
Well, yeah, but that would.
But I think the reason that they specifically did that was because they isolated this big clump of genetic material, and they really just wanted to make sure that the protein and the RNA weren't doing the change, which is the same thing that we're doing within these mice, within the hypothetical mice, because we want to find out if it's the DNA, the RNA, or the protein.
Yeah.
Because it says specifically to prove that it's the DNA within the mice.
Don't die.
Did you guys see my cat walk in front of the screen? I was like, what are you doing?
My cat is enjoying my water.
Is that the last question?
Yeah. So I guess before we end, does anybody have any questions about this chapter?
Actually, I do have one question, and it's about it's about Avery, McLeod's, and McCarty's experiment. So, I guess at that point, had like scientists already determined what DNA was? Like they knew?
No. I'm pretty sure.
Or no?
Because that was like determining that DNA existed, like that there was a nucleic acid molecule called DNA was a whole set of different experiments, and
That's what I thought. They already knew what DNA was. They just didn't know if it was the genetic material for sure.
Mm-hmm.
So I think they knew what it was. They just didn't know what it did. Okay, that's what I thought because they had DNAs which they knew was an enzyme that degraded DNA.
Yeah,
But,
so I guess if they had DNA, they fundamentally had to know that DNA existed.
Yeah,
they knew it existed because that was my only question. Okay.
I was
more of a clarification.
When I was explaining the Hershey and Chase experiment, I called it a virus factory, not knowing that it was actually called a virus factor. And I was just making up a term, but I feel very proud of myself now for getting.
Wait, no, he said virus factory.
He said virus factor.
Factor. I'm pretty sure it was factor.
Factor is in here.
You'll probably stop recording now.
All right, so nobody has any more questions?
I think we're good.
I think we're good.
All right, awesome.
Awesome.
Chapter 11.
There you go. We helped. I can't speak.
All right.